Okay, so my name's Sarah. I'm a research associate and I work in the lab of John Laughlin, who spoke earlier today. And we're based in the Institute of Genetic Medicine, which is part of Newcastle University. But we're based down at the International Centre for Life by the train station. Sorry, the title of my talk today is Epigenetics and Osteoarthritis. So I'm going to spend a while explaining what epigenetics is and how we think this is related to the disease progression in osteoarthritis. So firstly, I'll start with genetics, and this is probably recapping a little bit of what Kat has just said. But cells are the fundamental working units of every human being, and the instructions inside every cell in our body are contained within the molecule called DNA. So as Kat explained, DNA is made of 4 different types of bases, and for ease we just term these A, C, G, and T. And there are 3 billion of these bases that make up the sequence of an entire human genome. Our genetic code, or the sequence of these bases in our DNA, determines the instructions that the DNA gives to our cells. And within this sequence, there are specific parts known as genes, and these provide the instructions to the cells on how to produce proteins. And proteins are the complex molecules that our body needs in order to function properly. And in the human genome, there are around 20,000 protein-coding genes. As John mentioned earlier, there are many different risk factors for osteoarthritis, such as age. However, we know that not all people, as they age, will develop osteoarthritis. Also, gender, we know that females are 3 times more likely to develop osteoarthritis than men, and other factors such as obesity and injury, which can damage cartilage and provide increased strain and stress upon our joints. However, we know that genetics contribute about 50% of the risk of developing osteoarthritis. And as John mentioned earlier, it's not a single gene that you inherit from your parents, but actually many different genes that work in combination. And once you cross the susceptibility threshold, will lead you down a pathway to developing osteoarthritis. This diagram is a human karyotype. So simply this represents the 23 chromosomes that we all carry. And to date, using the large-scale genome-wide studies that John mentioned earlier, we've identified 30 genes that increase the risk of developing osteoarthritis. And this diagram is just here to show where these are located throughout the genome. I'm going to talk to you later on in this presentation about one particular risk gene, and this is known as RUNX2. I've just labelled this here to show you that this is located on chromosome 6. And also, I'm talking about this today because this is the gene that I work on personally in the lab. But also, this was a particularly compelling candidate for being involved in osteoarthritis because we know that the protein that RUNX2 gene tells our cells to make is essential for the healthy development of the skeleton, so of cartilage and for bone. Okay, so moving on to epigenetics. So epigenetics are modifications to our DNA that don't change our genetic code. So these are changes in DNA that don't alter the absolute base sequence of A, T, C, and G. But they're ways in which our cells can control the expression of genes. So our cells use epigenetics to switch genes on or switch genes off. And this is really important in different situations, such as during development, your body might need to switch genes on that aren't needed once we reach maturity, and also in different cell types to make cells what they are. So maybe a cell in your cartilage expresses different genes to a liver cell or a cell in your heart. We also know that in certain diseases, various genes will be switched to the wrong state. So genes can be switched off, for example, when they should be switched on. And because we know that epigenetics are reversible, if we can harness this mechanism and use it ourselves, we can theoretically cure diseases in which epigenetics has gone wrong, such as cancer, aging, obesity, and potentially osteoarthritis. So what we want to know is whether osteoarthritis is caused by epigenetic modifications to DNA. There are different types of epigenetic modification, but I'm just going to talk you through one of them, which I'm going to focus on in my talk today, and this is DNA methylation. So as I mentioned earlier, DNA is made of 4 bases, which are C, G, A, and T. And what we do know is that in the DNA sequence, where you have a C and a G next to each other, the cells are able to add these small groups, so I've shown them as purple circles here, which are methyl groups, they're able to stick these methyl groups to the DNA, but only where there's a C and a G next to each other. And this is known as a CG site. And this DNA methylation is one of the ways in which our cells can switch genes on or off. So our aim in the lab is to understand the function of these genes that have been shown to increase risk of osteoarthritis and understand exactly how they do cause osteoarthritis and whether this is via epigenetic mechanisms. So this is where it starts to get a bit complicated because I'm going to talk about the project that I'm working on, but I will try and explain it to you if you bear with me. So as I mentioned, the gene that I work on is called RUNX2. So this is the gene that our bodies need to express in order for you to develop a healthy skeleton, so for formation of bone and cartilage. As we've mentioned, when we do these large-scale genetic studies, we look for changes in the DNA. So for example here, an A base at the top, which is found in most individuals, can sometimes be changed to a G base. And while this is found in a large number of members of the healthy population, we find that this occurs more commonly in people who do develop osteoarthritis. And what we also found when we looked at this region is that this change is not within, but very near to the RUNX2 gene. We also realized that very close to both this change and the gene is what we call a CG island. So this is where there are lots of these CG sites that can be methylated very close together. And this analysis led us to ask two different questions. So firstly, we wanted to know, does carrying an A or a G affect how methylated this CG region is, this CG island in your DNA? And secondly, we wanted to know whether methylation in the CG island could switch the RUNX2 gene on or off. So to do this, we use the cartilage samples that Ioana collects from the patients undergoing total hip or knee replacement, and we extract the DNA. And we can then measure the amount by which this DNA is methylated. And we do this using a technique called pyrosequencing. So we do this in the lab on a machine called a pyrosequencer, and it gives us an output that looks a little bit like this on the left. So for each individual CG, it tells us that in this one particular patient sample, their DNA was 95% methylated. What we wanted to know, however, was whether carrying an A or a G would affect methylation. So what we look at, and each of these points on this graph represents a different patient sample. So we repeat this measurement in hundreds of patient samples. And what we can see is that having a G, which increases the risk of developing osteoarthritis, is also associated with having lower levels of methylation in the CG island. So the second question that we wanted to know was whether ultimately this CG island was being used by the cells to switch the RUNX2 gene on or off. And as Ioana mentioned before, we can grow human chondrocytes or cartilage cells in these flasks. And we use the technique to edit the genome that John briefly touched upon earlier, which is the CRISPR-Cas9 system. And this is essentially a pair of molecular scissors. So we can use this system and introduce it to these cartilage cells. And these molecular scissors will cut this region out of the DNA of each individual cell within this flask. And we can then measure the effect this has upon the RUNX2 gene. And what we found is that when we did delete this region in all of the cells, is that we saw that a switching on of the RUNX2 gene. And this tells us that, yes, actually this region that can be methylated is being used by the chondrocytes to switch the RUNX2 gene on or off. So in conclusion, what does this data tell us? Well, we know that this genotype, so if you're an A or a G, at the change that increases the risk of developing osteoarthritis, is correlated with methylation of DNA at a nearby CG island. We've also carried out functional work and seen that actually changes in methylation in this CG island are used by your cartilage cells to switch these genes on or off. Moving on from this, we can carry out genome editing further to alter this region in chondrocytes that have been damaged and see whether we can cause normal function to return. And together these techniques help us to really understand how these changes in our DNA do cause osteoarthritis. And secondly, it will enable us to modify damaged cartilage to produce more resilient cartilage and hopefully introduce this back into patients with osteoarthritis. Thanks.